and all those things, you know. So I think all of us were full with that. So I kept the lecture again very basic to tell the student that we are talking so much of immunology, what it is, what kind of a system is there in the body which is giving us protection. Though we say it sounds very simple, immunity, but it is a very complex mechanism, like I said. So we have all these micro, you can see in this slide, we have bacteria, we have fungi in the environment, we have protozoan parasites, we have helminthic parasites, all types of microorganisms are present in the environment. But we do not suffer from all the diseases all the time. And with COVID-19, you see so many people, they were positive, but they never got the infection, not they never got the symptoms, you know. So everybody said because they have a good immunity, and that is why they are not getting the infection. And others, though they appear to be very strong, but we had so many fatal cases, you know, even in the university, who succumbed to these kind of infection. So it is important to understand what is giving us this protection, how the body is protected from this. So before going, just for the students, I had kept few definitions. What is immunology? We said immunology basic we are going to study, but we should know what is immunology. It is science that. Now, what is immune system? Immune system is the system which is giving us protection from different types of diseases which are present in the environment. And it is so adaptable that, like you see, the virus is very small, you cannot see, but you are protected. It may be in nanometers, like polio virus, it is only approximately 30 nanometers, you are protected. You are also protected to a very, the largest, this giant kidney, one example I have given, kidney worm, you are protected against. So you can see how diverse the immune system is, that it is able to protect you from the smallest of the organisms to a very large organism. And what is immunity? Then immunity is derived from a Latin word immunity, exemption. When we say exemption, that means you are resistant to so many different types of infectious diseases to which you are exposed day to day, but you do not develop these infections. So we have three terms. We have immunology, we have immune system, and we have immunity. So before going into any subject, that is my always, I like to teach history of a particular subject so that we appreciate. Now we know word immunity, now we know we have T cells, B cells, we know antibodies, everything we know. But we need to appreciate the people who have been involved in giving this kind of science to us. So briefly, I will just go through the history that immunology science developed very slowly from olden times itself, ancient times, where people just made an observation that if you get some kind of infection, so in those times, in ancient times, people from plague, they should used to die from any kind of infectious disease, you know, flu, because no treatment was available other than whatever Ayurveda, different types of alternative therapies were available at that time. So they just made an observation that once a person is cured from any kind of infectious disease, the person does not get the infection for the second time. And this was just an observation, but it is there in the literature that there was a great historian who had put in on records that people the sick, you know, that means that they knew that once a person suffers, gets the infection of plague, people used to die, we know. But those who survived the plague, they were then able to nurse the other people who were suffering from plague. And nicely they could do it because, again, plague is very contagious disease. And these people could look after them, you know. So after that, also these observations were there, but 2000 years passed by, and no, within 2000 years, and you will agree with me that Chinese and Turkish people, they are actually much advanced. They like to try different things. So in 15th century, Chinese and Turks, what they did, and another deadly disease of those times was smallpox. So what they did was, you know, when smallpox is there, initially the pustules have some kind of a fluid in them. when the after 10 days they start drying up. So there is a crust is formed on the this smallpox pustule. So what they did was they took the crust of the smallpox pustule, made cut in the body of the people, and try to insert this crust into the skin, or they asked the people to inhale. And they found that some people who were taking this kind of treatment, they were getting immune to this infection. They never got. So this method was called as variolation, and this was used first time for smallpox. It was so popular in those times that in 1718 also, wife of a British ambassador, she tried it on her own children, and she could protect her children against these diseases. So this was a first attempt of immunization. What we say immunization, that is, we are protected against these diseases. Now, Edward Jenner name, I think all of us are very well aware with Edward Jenner. He was a physician. He also made an observation that there's some people who suffer, who are working with the these cows, जो दूध वगैरह निकालते हैं. So they observed that all these milkmaids who were working with these cows, they were milking the cows, cowpox. So all these milkmaids, they never suffered from smallpox. So he was wondering why this is happening. And then he found that these cowpox is a very mild disease, but they were protected because of cowpox. They were protected from smallpox. So there was again another he thought, let me make a try, and he inoculated this fluid from cowpox into an 8-year-old boy. See, it was at that time these ethical guidelines were not available. Like, if you want to do such things now, you cannot do because of ethical reasons. But he tried this at that time, and then he injected this animal with the smallpox, and he found that the child was protected from smallpox. So this was a really great beginning for the science of immunology. So it spread through Europe, but even again after that, 100 years went by, but nothing much happened during that time. So then after Jenner, Louis Pasteur, he was trying to do some work in his laboratory where he was working on foul cholera bacteria. He found that there are bacteria. He cultured the bacteria in the laboratory, and he found that cholera in the fowl. So serendipity is one which actually always is a helps in advancement of science, and same thing happened in the case of immunology also. Cholera bacteria, and he wanted to use this foul cholera bacteria, infect the chicken, and then there was summer vacation, and he went on a holiday. When he came back, then he used that culture, which bacterial culture, which was grown for a long time. So he tried to infect the chicken with this foul cholera bacteria, and he found that these fowl, they never developed any disease. He was very surprised that why these chicken are not getting this infection. Then what he did, he again grew the culture, and then he wanted to infect the fresh chicken, but it so happened that the fresh chicken were not available. So he used the old chicken only, old fowl, which were cured of this infection, which was given infection with the previously. To his surprise, these fowl again did not get infection. So he found out that if you grow the bacterial culture or any culture for a prolonged period of time, it weakens the virulence of the pathogen, and you could use this as a vaccine. So this was a major advancement in science, which was done by Louis Pasteur, and to honor Jenner, because Jenner was the first one who had given cowpox pustule fluid into a child, but that was not so popular. So he wanted to honor Jenner for this, and he named this technique of injecting the attenuated strain, using the attenuated strain as a vaccine. He termed this as Vaxa, and from there only the vaccination word has come. So Vaxa was just to honor Jenner's work. So I hope it is clear. So this are the landmark beginnings in the history of immunology. That is the work of Jenner with the cowpox and smallpox. He found that the attenuated strains, aging weakens the strain of or the virulence of a pathogen, and then this attenuated strain then could be administered as a vaccine. Then he tried to extend this to other diseases, and he found that he did these experiments in heat attenuated anthrax bacillus. Then he took two group of sheep. In one group, he challenged them with this vaccine, heat attenuated anthrax bacteria, and in other was the unvaccinated group, and all the vaccinated group, when they were challenged with the live vaccine, all vaccinated lived and all the unvaccinated they died. So these experiments marked the beginning of immunology. And in 1885, another boy who's very famous in immunology is Joseph Meester. This boy, he was bitten by a very badly, he was bitten by a rabid dog. So in that time, because of this, he must have been dead, you know. So two physicians, they approached Pasteur, and then Pasteur had did rabies preparation, and he was preparing it in the rabbits. So he took the serum of this Joseph Meester, and he survived. So this was another landmark in the history of immunology, where a boy was, you know, saved because of this rabid infection also. So this boy Joseph Meester, he was then custodian of the Pasteur Institute, and there is some interesting history also, that during the World War, when Germans wanted to come and take these Pasteur crypts. Actually, this Pasteur had kept lot of, you know, virulent strains of the pathogens. So Germans wanted to come and capture this and spread all these diseases in the population. But Joseph Meester, he gave his life, and but he never gave the possession of these Pasteur crypt to the Germans. So this is a very important young boy. He died also very early, but he was custodian at the Pasteur Institute. He died in 1940. So then with these, whatever observations were made, the experiments were made by Louis Pasteur as well as um, Jenner. People started thinking after that, that what is present in the body which is actually giving you this kind of a response. You know, you are injecting the pathogen, attenuating the strain, but what is happening in this field, and there were two important people, von Behring and Kitasato. They wanted to study the insights of mechanism of immunity. So what they did was they isolated serum, either coagulated blood, or if you add the anticoagulants, then you can separate out the serum or the plasma. So they tried to use this plasma, and they injected it into the animals. They took serum from infected animals, injected it into the other animals, and when they tried to give the infection, they found that serum contains something which is giving actually immunity to a particular animal. They saw that this serum could actually neutralize toxins, it could precipitate toxins, it could rupture bacteria. They thought that serum contains many components, each one is giving different kind of activity. It can be, it has antitoxin activity, it has precipitin, you know, agglutinin activity, different types of activities were there. But later on, it was found that all these are actually because immunoglobulins, or what we call the gamma globulins. Studies were done, and they could isolate the gamma globulin fraction, and then antibodies were found. Now, since this was present in the fluid, that is a serum. Serum is a fluid part of the blood, and so this, they called it as humoral immunity. From there, the term humoral immunity is come. That is the immunity which is mediated by the antibodies, and it is contained in the body fluids. So you call it as humors or humoral immunity. So I think all of us are well aware what is humoral immunity and what is a cell-mediated immunity. So this is how we got the term humoral immunity. Now, till today also, you are well aware that whenever you have any snake bite, you have any scorpion bite, so how do you get? You take some kind of vaccines. What are these vaccines? They are nothing but the hyperimmune sera, which is isolated from these animals, especially horse and sheep. So what you do, you give small doses of these venoms, either snake venom or scorpion venom, into these animals, and slowly they produce antibodies against these venom. So these antibodies, we call them as hyperimmune sera. So whenever any person has a snake bite or a scorpion bite, if you give hyperimmune sera, then it neutralizes the toxins. They are called as neutralizing antibodies. It neutralizes the toxins so that the person is protected from this whatever pathogenicity this venom is going to cause. So even today, this kind of treatment is there for diphtheria. We have for tetanus. We have for different types of bites from these deadly animals. So in the parallelly, there were other people in the serum, and tried to do a lot of work on cells. They isolated the WBCs, white blood cells, from the blood of animals, and they tried to do if you take WBC, they were having more phagocytic ability. That is, they were able to kill more pathogens. So that is where the role of these WBCs came in. Some experiments were carried out by at Rockefeller Institute by Merrill Chase and others. They transferred these WBCs between guinea pigs, and finally, the net result was that they said that no, it is not the serum which are responsible for transmitting the immunity. So since they found they gave the name cell-mediated immunity. So there was a big fight going on between these two groups. One was insisting that cells are responsible for transferring immunity. On the other hand, the others said, no, it is a serum. So it was Bruce Glick of Mississippi State University. He then said that there are two types of lymphocytes. We have the T cells from the thymus, which are responsible for cell-mediated immunity, and you have B cells, which are from the bursa of Fabricius in birds, otherwise bone marrow in adults, which are actually responsible for the humoral immunity. So, and he also said that both immunity are have to go hand in hand, to give you protection against different perspective, how people made observations, types of observations, how Jenner carried out the experiments, how Louis Pasteur did very beautiful experiments, and then you had people who got then later on Nobel Prize to define that it is a serum as well as the cells which are responsible for transferring a protection against different types of diseases. Now, coming to the immune system. Now, we know that in the immune system, we have two types of immunity. All of us have been studying from, I think, from lower classes itself, we start studying innate immunity and adaptive immunity. When we talk of innate immunity, so first barriers, if any pathogen comes, what is the first barrier? The first barrier will be your skin, because your skin has epidermis layer, is closely can and kind of insect bite. Only then when it bites, it will directly enter into the blood and cause infection. Otherwise, skin gives you a lot of protection. Other than that, you have the gastrointestinal tract, you have respiratory tract, you have urogenital tract where the opening is there. All these openings are protected with a mucous membrane. A lot of mucus is present. If any kind of pathogen enters, then that pathogen is entrapped in the mucus and then it is thrown out. Or you have nasal hair. So these are the external barriers. if you look at the internal barriers, now in the skin also important is that, you know, the skin, the pH of the skin is maintained between 3 to 5. pH is 3 to 5 only because of the presence of sebum, which is secreted by the sebaceous glands. Now, because of this pH, most of the pathogens, though they come and sit on your skin, but they are not able to cause any infection, or they are not able to multiply because of the kind of pH that is present on the skin. Now, then you have the internal barriers. Now, the internal barriers, you have the first line of defense and second line of defense. First line of defense, I think all of you have also must have heard about the phagocytes. Anyway, we are going to talk in details about the phagocytes. We are going to talk about the natural killer cells. Then we have second line of defense, which includes actual immunology, that is the T cells, B cells, which form part of the adaptive immunity. Now, what is the difference between innate and adaptive immunity? When we say innate, the term itself says that it is the first line of defense and it is inborn. Inborn means right from birth, this innate immunity is present. Even before the exposure of to any kind of antigen, this immunity is there. Right? You have skin, you have inflammatory response, everything is present, and it is very non-specific. Any pathogen tries to enter, it is going to protect you against this kind of pathogen. When we talk of adaptive immunity is highly specific, and it develops after exposure to an antigen. Once a pathogen enters into a body, after that an infection is caused, or it is presented to the immune system, to the T cells, B cells, and then second time, and you will have memory for this kind of infection. Memory is there, memory cells will be there. You will have T memory cells, you have the B memory cells. So whenever second time that antigen comes into your body, in case of innate immune response, there is no memory. Second time, if same pathogen comes, innate immune response will again start afresh, will take that as a new entry into the body. But whenever adaptive immune response is there, it will recognize that pathogen, that this pathogen the body, and immediately the memory cells will come into play, and it will be much more rapid response. Adaptive immune response is much more rapid, and whereas this innate immune response is not that rapid. This is for limited period, this is highly diverse, and lot of memory is there. That is the difference between, and in this you have major components of innate immune response are the skin, the phagocytes, and in this you have, I like always said, the T cells and the B cells. Now, what is happening in the innate immunity? Once the microbe enters, for example, there is some cut in the skin, and whenever you have any injury, naturally there will be cut, blood will ooze out of this, microbe will enter. Once the microbe enters, it will be taken care by the macrophages. Now, what does the macrophage do? Macrophage has a phagocytic ability. It will phagocytose the pathogen, and either into, because pathogen, this macrophage will have lysosomal enzymes. There will be phagosome will be formed, phagosome lysosomal fusion will be there, and after fusion it will break up into different parts. On the other hand, some part of this pathogen will also be presented on the surface of the antigen presenting cell. This is called as an epitope. So this epitope will be presented by this macrophage to the different types of cells. Now these cells can be presented to the T cells, it can be presented further to the B cells. So this is innate immunity, this is adaptive immunity.
Last बस इसके बाद इसकी कोई नहीं last
When we talk of components of immunity, like I said, innate immunity is a natural immunity present from birth. It is first line of defense. It is present before the onset of infection. It includes non-specific elements, and it contains a set of disease resistance mechanisms that are not specific to a particular pathogen, but includes cellular and molecular components that recognize classes of molecules peculiar to frequently encountered pathogens. I will not be talking about the PEMPs and DMPs here, because that itself is again a very large topic. Now, if we talk of acquired immunity, simplicity and remarkable memory, response is stronger and more effective, and the ones cellular agents which are responsible are the lymphocytes and the antibodies and other molecules. Now this figure shows the in the innate immunity we have the anatomical barriers. Now anatomical barriers, I think I have already explained, that skin which has the sebaceous glands, you have the mucus which will push most of the pathogens out, you will have the flushing action of saliva, tears and urine. In saliva also you have different types of enzymes which will kill the pathogen. So this is a diagram for that. Then in the intestine, you have the acidity, and HCl in the stomach almost kills whatever different pathogens which enter into your body. Now, next important part is the the for example, there is a tissue damage here, and whenever tissue damage is there, the bacteria will enter into the area wherever the damage has occurred. Immediately bacteria will occur, and you have seen that whenever you have any injury, after few hours you will have a pus formation, you have redness of that area, it will be the temperature will be increased, it will be hot, swelling will be there. All these events we have already noticed. All of us have some kind of injury at some time in the life. So these events, what inflammatory response. Now, what happens during an inflammatory response is whenever this injury occurs, bacteria enter. So the body will respond in such a way that there will be all these WBCs, they will start coming near the area of. So this is called as margination. These capillaries will increase. The WBCs will have margination. They will adhere to this endothelial wall, and slowly then they extravasate here. And they will move into this area, and they will try to go towards the place where there is bacteria is present. Why phagocytes are going? Because the name phagocytes means they are going to phagocytose these bacteria. So they will reach here and they will engulf the bacteria here. I have a better figure. I'll just explain here. So whenever injury occurs, you can see the thickness of this artery is very less. But in the second step, you see it has increased. So this increases for whenever this increases there, that is why you have a swelling. More of RBCs come into the place, then you have a lot of phagocytes. So you have a swelling, temperature of this area is increased, and edema is there, redness is there whenever inflammatory response are there. So whenever injury occurs, chemical signals are released by these macrophages and mast cells, and then they move towards the nearby capillaries. Now you can see here that antimicrobial proteins, fluid and clotting elements move from blood to the site of the infection, and slowly the blood clot will be formed. Now you see many bacteria are there, but in the next step you will see, and many of the chemoattractants are, and then you see here that the bacteria are being engulfed by these either the neutrophils or the macrophages, whatever reaches the site, and this is what we call it as inflammatory response. So whenever occur, that is a positive response, actually protecting you from different types into the body. The acidic environment on the skin, which I told you, pH is 3 to 5. Then the mucous membranes, which have mucus, which entrap the foreign organisms. Then the cilia propel the microorganisms out of the body. Then another thing you must have come across is that there is, whenever we take antibiotics, after that we have some kind of problem in digestion. Why it occurs? Because antibiotics, they kill the normal flora also, which is present in the intestine. Now, this normal microflora is very important for digestion, as well as since the microflora, this normal microflora is present, it actually does not allow the microbes to have attachment sites on the gastrointestinal tract, because they compete with the pathogen for the attachment site. So this normal flora is very important as far as the gastrointestinal tract is. जो आप ये योकुल्ट और ये सब दही सब खाते हो, that contains all these normal microflora only, which actually helps you for different types of mechanisms in the body, especially the digestion. Then the physiological barriers are there. For example, normal body temperature, which inhibits growth of the pathogen in the body. Whenever any pathogen enters, fever is there, so that the pathogen is killed because of the increased temperature of the body. Then acidity in the stomach kills most of the microorganisms. There are some chemical mediators like lysozymes, interferon, complement, toll-like receptors, collectins. All these I will not go into details. Then you have the phagocytic, endocytic barriers. I already told you how the phagocytes, they engulf these bacteria either through phagocytosis or endocytosis and kill these pathogens, which I have just explained. Now, another important thing to understand is, or to know is for the students is, whether the invertebrate animals, the higher plants, do they have this immunity? Do they have innate immunity? Do they have adaptive immunity? So you will see here, this figure nicely describes that innate immunity is present in right from higher plants to invertebrates to the vertebrate animals, but adaptive immunity is not present in animals. Now, so all these lower animals, they have some kind of phagocytosis, they have some antimicrobial peptides, they have some pattern recognition receptors, everything is there. But if you see the adaptive immunity, which includes the T and B cells, they start only from the vertebrate animals. Antibodies and everything is there only from the vertebrate animals. Now, I've just put in few questions. Why do we need immune response? Why do we need to study immunology? Where this immune response is located? We need to know the answer to. I said this is a very flowery term, immune system, immunity. You know, everybody talks about this, but actually most of the people do not know the importance of immunity. Now I think with COVID-19, we are little bit, more aware about what is the importance of immunity. Otherwise, we were really ignorant. Why do we need it? We need it because there are so many pathogens in the environment. Like I said in the beginning, you have bacteria, you have viruses, you have parasites, you have dust, you have fungi, so many things are there. Then you have cancer, you have tumors, all these are there. So we need to protect our body against all these different types of infections. So what is protecting you? It is your immune response, which is protecting you against these diseases. Why do we need to study? So we need to know what is allergy, we need to know what is inflammation, how these autoimmune disorders are occurring, what is organ transplantation, what are these different types of pathogenic diseases which occur in the human, and how our body is protecting us against different types of diseases. Where it is located, how it works, where it is located, like your principal also said, that it is the defense system. Defense system, what it has an army, it has weapons, there is a defense organization. Then what next is required? You need to communicate, and there is a lot of cooperation is required, different sections. Only then you will have enemy free land and barrier. So what is the army in this? You have different types of cells which are present. Army and weapons are your cells. How they are organized? Everything is organized, interlinked, which I'll show you, with each other. And like I said, cooperation between innate and adaptive immunity is required. Cooperation between cell mediated and humoral immunity is required. Only then you will have a disease-free body. So all this system, we will slowly see what kind of system is present. We said we have different types of cells which are responsible for different types of immune responses. So from where these cells are coming? Now these cells are coming from, now these cells are coming from there is a process which we call as hematopoiesis. All of you must have studied about hematopoiesis. It is the process of formation and development of blood cells. So you have what you call it as hematopoietic stem cell. Stem cell again is a very fashionable word. All of us know what is a stem cell because stem cell has the ability to self-renew. It can differentiate into any other type of cell. That is what we call, that is why we call it as a stem cell. So hematopoietic stem cell will further can differentiate into different types of cells. First, it will form the progenitor cell. So this is a pluripotent cell. Further, it will differentiate into progenitor. Then they cannot go back differentiate into any other type of cell. When we use progenitor, that means now they are defined to become this type of cell. So you have here pluripotent cell, but here they are called as progenitor cells. Now from these hematopoietic stem cells, you have two types of progenitors. One is a myeloid progenitor and other is a lymphoid progenitor. Now the myeloid progenitor will give rise to different types of other progenitors, like it will give rise to granulocyte monocyte progenitor, eosinophil progenitor, basophil progenitor, megakaryocyte, erythrocyte, which is further giving rise to neutrophils, monocytes, macrophages, basophil platelets, and so on. On the other hand, if you see this lymphoid progenitor, this will give rise to the natural killer cell. This will give rise to T cell progenitor, will give rise to T helper cell, T cytotoxic cell, B cell will give rise. Some of them will give rise to natural killer cells and also the dendritic cells. So all these cells will come from hematopoietic stem cells. Now we take these cells one by one. So, for example, you have four main types of cells which originate from the myeloid progenitor. Sorry. So one is the RBCs. All of us know about red blood cells. So you have basically two types of cells, red blood cells and the white blood cells. Now in white blood cells, we have monocytes, we have granulocytes, we have megakaryocytes. Megakaryocytes give rise to the platelets. Importance of platelets also, I think all of us know very well because of this dengue, during which most of the viral infections, the number of platelets, they significantly reduce, and then a lot of transfusion of these platelets has to possible for clotting of blood during injury and loss of blood. Then another important cell is the monocytes. Now, when the monocytes are circulating in the blood, they are called as monocytes. Once these monocytes go and settle down in the tissues, then they are called as the macrophages. You see the difference between monocyte and macrophage. This number of pseudopodia, these processes, they increase. Number of lysosomes also increase in the macrophages. The tissues, they have their own terminology. If they settle down in lungs, they will be called alveolar macrophages. If in the bone, osteoclast. In connective tissue, histiocyte. In intestinal, macrophages. In the brain, they will be called microglia. In liver, they will be called as the Kupffer cells. So then we were talking of the white blood cells. Like I said, we have two types of these cells. One is red blood cells and the white blood cells. White blood cells, you can further differentiate into granulocytes and the agranulocytes. In their cytoplasm, you call them as granulocytes, and you have in these three types, neutrophils, eosinophils, and in the agranulocytes, monocytes and the lymphocytes. This is what I was telling, the plasma and the serum. If you coagulate the blood, you separate out the liquid part, that is called as serum. And if you add an anticoagulant and then do centrifugation and settle down, then you call this as plasma. And there are different steps where you can separate out these cells by doing density centrifugation. This out of this plasma. So the percentages of neutrophils, if you see, well, you must have all sometimes got your blood test done. So this is the kind of report you get. The number percentages of these normal cells. Neutrophils, you need see the maximum number of cells, percentages of cells are the neutrophils, around 50 to 70% of the total cells. Lymphocytes are 20 to 40%, monocytes 1 to 6%, eosinophil 1 to 3%, and basophils are less than 1%. One by one, in the innate immune system, and they are the front line of attack during an immune response. And you can differentiate on the basis of the cell staining. So we show how, and we make a blood smear, fix it, and then stain. And this is how you see the slide. These are granules. That is why it is called as a granulocyte. You see so many granules are there which stain with the eosin dye, eosin. So that is why you see these pink granules. It constitutes 1 to 3% of the circulating leukocytes. Now leukocytes are nothing but the white blood cells. They play an important role in defense against multicellular parasitic organisms. Like I've shown one example. When this infection is there, then the eosinophils, they come and bind. So this through one which is called as IgE antibody portion, the pathogen is entering attached on this side. FC portion, eosinophils are there. Once they bind, then they release all these pharmacological, how then it causes lysis of the pathogen and it helps in elimination of the pathogen. So that is the role of eosinophils. The neutrophils, I said they have almost 40 to 70% are there. So they are the most numerous of the populations. They have multi-lobe, 4 lobes, 5 lobes, and so on. Their main role is in inflammation. I showed you at the inflammatory site, the main cells that come at the inflammatory site are the neutrophils and the macrophages. And now next are the basophils of these granulocytes. They also have a bilobed nucleus, but S-shaped type of nucleus is there, and they have these again lot of granules there. So these granules are stained with the basic dye, that is a methylene blue. So you see blue colored these granules here. And they are, though their number is very low, but they are very potent. Even the few cells that vessel allergic reactions, and they have high affinity for this IgE antibody, which I showed you in the previous slide. Another important cells which play a significant role in allergic reactions is the mast cells, and they are also found in a wide variety of tissues, including skin, connective tissues of various organs, respiratory, genitourinary, and digestive tracts. And these cells have large number of cytoplasmic granules which contain histamine, are the important ones in allergic reactions, leukotrienes, prostaglandins, etc. And like I said, they play an important role in the development of allergies. Now we come to the agranulocytes. So they are the lymphocytes. Among lymphocytes, you have the T cells, B cells, and the NK cells. There are some others also, but we will restrict for the beginners only with the T cells, B cells, and NK cells. Now the B cells, again, this is the surface antibody molecule you have studied. So you have two types of antibodies. You will have either the secretory antibody, which is found in the serum, or you will have the antibody which is bound onto the surface. Now, what is the purpose of this antibody here? This is the site on which the antigen is going to bind and activate the B cell. So this is called as antigen binding receptor. So this portion is called as FAB portion. This portion with which it is bound here, that is called as a. Lymphocytes, they were first time found in the birds in the bursa of Fabricius. Later on, that is how the name B lymphocyte came, but later on in the vertebrates also it is found in the bone marrow. So this B alphabet was retained, so they are called as B and display antibodies, and they also found antigen presenting cells. Why? Like I already binds onto the surface of these membrane antibodies which are present on the B lymphocytes, and they present antigens to the T helper cells. We will get to this later, and I think for you, I think this is sufficing. Now, then we have, this is the structure of the antibody. Like I said, it has two chains. This is the heavy chain and this is the light chain. We will not go into detail, but since we talk so much of antibody, you need to know structure. So like I said, this is a soluble antibody, is not attached to the cell. This is the surface antibody which is present on the surface of the B cells, which I showed you in the previous slide. So this is say the heavy chain, this is the light chain, and there are the disulfide bonds in this. So we will not go into detail of the T. Call them T cells because they are found in they mature in the thymus, and they express the T cell receptor on their surface. This is the T cell receptor which is present on their surface, and through this only they recognize the antigen. Some of the detail I will show you further. So they have two types of cells. They have the T helper cells and the T cytotoxic cells. Now, the main difference between B cells and T cells is that B cells can recognize when it binds onto the surface of this on the antibody molecule. But T cells cannot recognize the antigen unless it is presented along with the major histocompatibility complex. So this is the antigen presenting cell on the surface of which you will have the major histocompatibility complex. So this is the epitope which is being presented. So once it is presented through this MHC, then T cells on their surface, they have the T cell receptor through which this will then recognize. So this, since this is a CD4 cell or Th1 cell, so this will T helper cell. So this CD4 is surface, and the second cell is the cytotoxic cell. So this also presents the antigen or the epitope, sorry, through the MHC class 1 molecules, and this is again here TCR. So you see the way B cell was recognizing the antigen is entirely different from the way the T cells recognize the antigen. Whereas T cells will recognize the antigen only when it is present in molecules. The antigen only when it is presented along with MHC class 2 molecules, and T cytotoxic cells will recognize the antigen only when it is present class 1 molecule. So this is the basic mechanism which underlies immunology. Differences between how these antigens are recognized by the T cells as well as the B cells. It is very important to understand this. Now, next is important cells are the natural killer cells. The name itself suggests they are natural killer cells. They do not express, they do not resemble either the T cells or the B cells. They do not secrete antibodies. They do not have specificity. They do not have memory, but they play an important role in defense against virus infected and tumor cells. And these virus infected and They are their own cells, self cells, but they have been altered because they have been infected with the virus. Now, if you see here, when our body recognizes a B cell, when it recognizes, a natural killer cell, when it recognizes a normal cell, it says, hi, hello, it's okay. It is able to recognize that it is our own cell. But once this is infected with any kind of a virus or it becomes cancerous, then the natural able to recognize cell. That is how important the natural killer cell is. Now you can see here, for example, this is a natural killer cell. On its surface, it has a molecule which is called as a receptor CD16. So it can bind to the natural killer cell. It has, if it has MHC on its surface, it can recognize this. But if this cell becomes infected with the virus, the NK cell will not be able to recognize because the surface molecules will be different when the cell is infected with the virus. So you see, do not see MHC on its molecule. So immediately natural killer cell will recognize that there are some cell surface proteins which are missing, and then it will try to kill. Proteases, they produce perforins. So all these perforins will kill the cell itself, and along with the cell, the pathogen will also be killed. So that is how important the can also kill through the antibody, and then when it kills through the antibody, we call it as cytotoxicity. Like I said, we have studied about innate immunity, we have studied about adaptive immunity, but I said in the beginning that it is interaction between the innate and adaptive immunity. So how do they interact? They will interact with two methods. They will have cell to cell contact, and there will be some soluble messengers, like you have studied about hormones. Same way in the immune system, cytokines. There are some chemokines. So the function of the cytokines and chemokines are that they have chemotactic activity. They will, for example, in the inflammatory response, we said some chemotactic factors are released, which will invite all these neutrophils, for example. So macrophages will come to the site. Neutrophil chemotactic activity will be released. So neutrophils will come to the site. So this way, all these cytokines, chemokines, they help in recruiting the different types of cells at a particular part so that they perform their function. So what are these cytokines? They basically act only on the cells which bear receptors. ऐसे नहीं है कि they cells they produce cytokines and on itself they will show an action. For this cell to this cell, there has to be this cytokine. There has to be receptor on the other cell, only then that will be recognized. So there is receptor mediated recognition is there. I'm just taking it briefly so that we don't miss any part in the whole cycle of the immunity. I didn't want to teach cytokines, but I thought otherwise you will not know how the coordination is taking place between different kinds of cells, So different activation of different types of cells, they inhibit, they also help in chemoattractant. So briefly I will show you the kind of cytokine only in the just for few cells I have shown. For example, for these stem cells, we said from hematopoiesis into different types of cells. So for the differentiation of these different types of cells, you see you will need so many different types of cytokines. These are interleukins, these are granulocyte macrophage colony stimulating factor, this is TNF, this is macrophage colony stimulating factor, interleukin 1, 6, 12, TNF alpha. You see everywhere. For transformation of each cell into another cell, there are so many different types of factors, chemotactic factors, there are so many different types of cytokines are there. are important. So without cytokines, nothing can take place. Nothing in immunology will happen till the time a particular cytokine is produced. I'm not going into details. I just showed this figure to show you that there are something called as cytokines which are actually responsible for the kind of coordination which will take place between the innate and adaptive immunity, between the humoral as well as the cell mediated immunity. Now, where the immune system is located, we have different types of, we define them into different types of organs. We say we have primary lymphoid organs and we have secondary lymphoid organs. Primary lymphoid organs, in this we have bone marrow and thymus. And why we call them as primary lymphoid organs? Because they are the maturation site where these cells are produced. Cells are produced in the bone marrow and they mature there. T cells are produced in the thymus and mature there. So we organs. Now move towards the different organs such as spleen. They move towards gut. They move towards different mucosa. So when they move towards these adenoids, primary lymphoid organs. So we will briefly take them. Now, so primary lymphoid organ, I had just put down a slide where the immature lymphocytes develop and organs where differentiation competent cells takes place. This in marrow, it is where antigen is localized so that it can be effectively exposed to mature lymphocytes and initiate the immune response. This includes spleen, lymph nodes, tonsils, appendix, Peyer's patches, etc. Now, primary lymphoid organs, well, first we take up the thymus. Now, this thymus is, if this is your heart, this is where it is located. It is a bilobed organ which is located just above the heart. Now, each lobule is bound by a capsule. You can see here, this is a capsule. And each capsule is separated by connective tissue, which is called as trabeculae. These are the trabeculae. Capsule. Now, we will not go into detail of these. So we call them as T cells. Now, the thymus, the size increases till puberty, and after that it atrophies. It is important for the development of immune system at an early stage, and thymectomy at early stage results in less number of T cells, and person may suffer from different types of T cell related diseases. Bone marrow, it is located in the middle of the bones, especially your vertebrae, pelvic and leg bones. Your long bones, they are the main ones where these B cells are found. It generates T cells, B cells and macrophages, cells that travel. Now coming to the secondary lymphoid organ, the lymph nodes. Whenever you have an infection, you must have observed, this must be general observation, that the doctor will check your neck or the armpits for some kind of a swelling. So whenever they see that, that is basically to this thing is there. Swelling generally occurs in the lymph nodes. Lymph nodes are again an important immune system. They drain the lymph fluid from the nearby organs of the body, and they are the first ones actually who filter your blood against different types of infections. Any infection enters, first it is at the lymph node, and then only after that it will be moving. Cortex, you have paracortex, you have the medulla part. Now, in the cortex, you have the B cells, macrophages, and the follicular dendritic cells. Dendritic cells are the antigen presenting cells. In the paracortex, you have T cells and the interdigitating dendritic cells. So, paracortex is the T cell rich area, cortex is a B cell rich area, and medulla mostly plasma cells are there. So, you see in this lymph nodes, you have T cells, you have B cells, and the T cells and B cells, so that further whatever action has to take place, development of adaptive immune response will take place. Recognize lymph. Now, spleen is located here. You can see in the left quadrant of the abdomen, and it is red in color, and red because the outer part is called as a red pulp. Now, red pulp is basically the one which destroys the defunct RBCs. They are also destroyed in the red pulp. Now, spleen is very important for filtration of the blood. Then you have the red pulp, and the inside you have a white pulp. Now, a white pulp is the ones, for example, this is splenic artery. So, surrounding the splenic artery, you have periarteriolar lymphoid sheath, which is called as pals. Now, this pals will be rich in T cells. Now, surrounding this, you have the marginal zone, then you have the primary follicle. Second primary follicle is the one which interacts with the any antigen which is presented by these interagitating dendritic cells. Further, when they mature, then they form the secondary lymphoid, secondary follicles also. So, the function of the spleen is to filter the blood and entrap the blood-borne antigens. So this was briefly about the secondary. There are many others: the mucosa associated lymphoid tissue, the cutaneous associated lymphoid tissue, but more are response, which includes the humoral as well as the cell-mediated immune response. So if you want to show that if any bacteria enters into the body, then what do you see here? That the phagocytes will engulf them. Either they will kill the bacteria, or it will also present this pathogen onto the surface, so that further the T cells will recognize this pathogen along with the MHC on its surface. It will then get activated, and then it can transform into two types of cells, either the cytotoxic T cell or the memory T cell. I said in adaptive immunity, each cell will be the one which is going to do the action against the bacteria, which is called as an. Now, in the case of the B cell, once they get activated after antigen interaction, they will get activated. They will also get differentiated into memory B cells and an antibody producing B cell. This is called as a plasma cell. Body will engulf the bacteria, and then phagocyte antigen antibody complex will be taken to the phagocyte to be eliminated. So this is the difference how it works. So this actually, both the cell mediated and the humoral immune response. For example, you have different types of pathogens. You have foreign proteins, you have viruses, bacteria. These are the pathogens to which your body is exposed. Now, once these pathogens enter into your body, so internalized antigen is digested by, and it is presented, it is broken down into different epitopes. Only the important are then which are immunogenic, they will be presented to the immune system. Like I said, if T cells have to recognize this antigen, it will be presented along with class 2 MHC molecules. So when it is presented with this, it will be recognized by the T helper cell. And what is recognizing here? You can see the major histocompatibility complex on this side. Sorry, the TCR on this side. So the epitope between the MHC as well as the TC. Now in the next step, T cell receptors recognize antigen bound to this, then it gets activated. Once it gets activated, then it starts producing the cytokines. Now these cytokines will contribute to the activation of the B cells. Cytokines will act. So here is the coordination between the cell-mediated immune response. So cytokines produced by T cells will activate the B cells, so that these B cells will then further produce the antibody. So this is link between T cells and B cells through the cytokines. Now, On the other hand, if antigen is presented through the MHC class 1 molecules, it will be recognized by cytotoxic T cells. They get activated and they transform into cytotoxic T lymphocytes. And these cytotoxic T lymphocytes can kill the altered cells, that is the tumor cells, cancer cells, or the virus infected cells. So cytotoxic T cells have a different role. T helper cells have a different role. Now these cytokines, when activates the B cells, then these B cells, they differentiate into the plasma cells as well as memory cells. So these plasma cells will produce antibodies against this pathogen. So antibody will bind onto this pathogen, its clearance from the body. What is the role of T MHC? What is the role of cell mediated as well as a humoral immunity? Now, what are the main characteristics of acquired immune response? We said initially we saw the difference, but most important aspect of adaptive immunity or acquired immunity is that it is very specific. First point, it memory, and fourth is diversity. Needs to remember these four important points of adaptive immunity: specificity, self non immunological memory, and diversity. I think we made very clear about specificity, that the immune system is able to recognize only those pathogens which are the T cells, B cells can recognize antigen. You have seen against which the receptors are there. Okay, self non-self recognition, it is able to recognize that it is a self cell and a non-self cell. Immunological memory, I already told that you have B memory cell, you have T memory cell. Diversity, I think I didn't mention that the immune response is so diverse that thousands, lakhs of different types of antigens are there, but the immune system is able to recognize all these pathogens, isn't it? It can recognize comes the immune so diverse, there is no pathogen or no antigen which the immune system is not able to recognize. And it includes two important types, that is the T presenting cells. Now, when any body and when a pathogen enters into the body, some experiment out where you see that if you give A into the body, so you have a response, antibody response, which starts on the 14 days. You call this as primary anti-A response. This we're talking of the. But when you give this same animal, if you inject antigen A, it will be secondary immune response. To antigen B, it will be the primary immune response. So you see, it will again the primary immune response, but secondary immune response appearing from day 6 onwards, much more heightened. So what is the difference between primary and secondary? That it starts much faster. So that way, adaptive immune response plays a major role in protecting against different types of pathogens against which you are going to be exposed for the second time. So these are again very important terms which you need to understand. What is a primary immune response? What is a secondary immune response? Then we also always talk of some natural immunity, passive immunity, artificial immunity. I thought I'll just define 2 or 3 terms. What is a natural immunity and what is a this partial immunity, and passive ko we call artificial immunity. What is active immunity? If host itself produces, how are you going to get this immunity? It can be either active artificial active immunity, or it can be natural active immunity. Artificial active immunity is immunity provided by natural infection. So I think it is very clear, otherwise we get confused between active and passive immunity, and what is artificial active and what is natural active. Passive immunity is that host does not produce antibodies on its own. You get it transferred from the other person produces antibody, you say you get it transferred into your body. It can be passive immunity or artificial passive. So wherever the IgG antibody produced in the mother crosses the placenta, as natural passive immunity, and the other one where we use vaccines like anti-venom and rabies vaccine, we call it as artificial passive immunity. I think I had put few slides how the immune response against bacteria occurs. It is basically phagocytosis. I will not go into detail. Phagocytosis and inflammatory response. This is how the pathogens are killed when they enter into the body with the help of these cells. They produce antibody as well as bacteria. Then again, phagocytosis response against fungi. Viruses also, they can be killed through innate immune response also through NK cells, and adaptive immune response through the activation of the cytotoxic T cells. Immune response against parasites is quite complex. We have some Th1, Th2 response, which I will not take. There are many, in spite of having a good immune system, we have a lot of immune dysfunction. Wherever there are some problems in the development of the immune system or any other things, lot of other diseases are there like allergies and asthma. I will not go into details. Transplantation and graft rejection, they themselves are one more topic, immunological basis of graft rejection, autoimmune diseases, and so on, autoimmunodeficiency disorders. So I'll just sum up that immune system we studied in this, the innate immunity, adaptive immunity or acquired immunity. Immunity, you have the physical barriers, then you have barriers like phagocytes. Adaptive immunity, you have the cell mediated, which you have the goal of the immune system is to cause the death of the cell, whether it is the innate immunity or it is the adaptive immunity. So it is I always like to show this slide where people, they worked so hard and got then after, Nobel Prize you get after maybe 20, 30 years or 50 years after you have finished your research. So these are some of the important people who are Nobel Prize winners for immunological research. We mentioned some names, Bering, Mechnikov, Karl Steiner, and so on. So all these are the persons who have won Nobel Prize. These are Kohler Milstein for the monoclonal antibodies and so on. And thank you so much. Taken more time, but I lost connection with the time itself. I'm sorry if I've exceeded time and made all of you sick. Thank you from my side.
Thank you, ma'am, for your very fruitful lecture. You have explained in detail, will be very helpful to our students. Now, I will request Dr. Hina to please ask the students for if any query and for vote of thanks. Dr. Hina.
Yes, sir. Now the session is open for queries. Audience can put their questions in the chat box. Ma'am, there is a question from Dr. Manish. He wants to know why there are less casualties in India due to COVID-19 as compared to rest of the world. Is this due to our strong immunity or due to other factors?
No, I think basically we have been talking about this for a long time. It is because we don't have that clean environment and we are exposed to different types of antigens all the time because we are not living in a very clean environment. So we are somewhere exposed to some kind of shared antigens of this virus, So we have already exposed. So whenever that virus enters, it is not taken as a very new antigen, and that is how the immune system is able to respond to that antigen. That is one. Another is that immunity part is again those people live in very clean environment, and you must have seen whenever any foreigners come to visit you, whenever they eat in your house, though I think we also have very clean environment, they suffer from diarrhea and different types of diseases. So immunity comes also with the kind of diet we take, like they are recommending lot of different type of kada and haldi, and you see every day diet we take this. Morning we'll have tulsi and adrak ki chai, then afternoon jo kitna khana banate hain we add haldi and jawan khate hain, all different types of takes. So both things are, I think, they go hand in hand. Sometimes somewhere we are more we are immune to these kinds of infections, and plus the dietary habits also have played a significant role. That is why we had less fatal rate was very less as compared to the other European as well as the Western world.
Thank you, ma'am. We don't have any other query. I think your informative and elaborative talk must have cleared the doubts.
No, no, I don't believe it. I think students would have asked some questions.
Yes, ma'am, there is a query.
Hmm.
My question is about this current topic, COVID-19. COVID acquired immunity potentially protect us from COVID-19, as according to study performed by scientists in UK, out of 60% of people demonstrated a potent antibody response, only 17% retained the same potency.
What is the question?
Shabnam Deep, the question is from Shabnam Deep. Shabnam Deep, you can unmute yourself and you can clear the question. Shabnam Deep, please unmute yourself. You can ask the
Yes,
question.
ma'am.
Yes.
Good afternoon, ma'am.
Good afternoon, beta.
Ma'am, my question is that I have recently read about this, that some scientists in UK have performed an study that showed that 60% of people who developed some antibodies against this COVID-19, only 17% of them retained. So is this possible for us to protect us from this disease, from acquired immunity?
Yeah, I think we also, for last 2-3 months, we also saw that antibody does not play a significant role in protection. There were so many antibody related tests which are being done, and so many people have antibody which I think they never got any infection also. And many people who were tested positive with the antibody, they also got the infection. So antibody that way did not play any significant role in protection against the infection. And I've already showed you in my presentation that in most of these infectious diseases, cell-mediated immune response is very important. So though antibodies, they are present and memory cells also are formed, but antibodies do not play a significant role in controlling against the infection, especially in case of COVID-19. Because most of the people, why this death was there, once the pathogen reaches the lung, there was lot of inflammatory response which occurred. Cytokine storm, what is called, I think आपने पढ़ा होगा अगर और भी पेपर पढ़े हैं, because of that cytokine storm इतना ज़्यादा inflammation हो जाता है, because of that a lot of whatever fatal cases were there, it was because of that. So antibody did not play much role in protection against COVID-19. That is
Yes,
why
ma'am.
you
Thank
will,
you.
in some patients 70% है, किसी में बाद में antibody, though memories, antibody does not stay. See, antibody will stay only for some time in the body. इसका life कितना है? Maybe few hours, 10-15 दिन है. After that, antibody will be, it will not be present in the body, because every time fresh antibody has to be produced by the B cells. They keep on producing antibody. So the moment the more antibody is not required, there is a negative regulatory mechanism which stops the activation of B cells, and no more antibody will be produced. But in the case of this, only when second infection comes, then the memory B cells will come into play. So antibodies do not remain for a long period of time in the system.
There is another query
Okay,
in
ma'am.
chat box. It is from Ria Thakur. Transplantation of tissues and organs often fails due to rejections by the patient's body. How is immune system responsible for such rejections?
Because immune system recognizes the transplant as a non-self. Immune system is so specific, it knows that this is not my body, not my organ, so it rejects it. So clearly I mentioned in that it's self non-self recognition of the immune system is very strong. So the moment there's pathogen, so whenever see these transplants are done for I think all their life they are on immunosuppressants. For their life they are on immunosuppressants. जो cyclosporin देते हैं, immunosuppressant देते हैं ताकि immune system उनका low profile रहे और immune system is not able to recognize that as, even if it is able to recognize, it does not generate lot of immune response, strong immune response to reject the graft because that graft is required for survival of the patient. But आपने देखा होगा जितने किडनी ट्रांसप्लांट के पेशेंट हैं वो हमेशा मास्क लगा के रखते हैं और बहुत ज़्यादा कंट्रोल्ड एनवायरनमेंट में रहते हैं. दे आर नॉट एक्सपोज्ड बिकॉज द मोमेंट दे आर एक्सपोज्ड टू डिफरेंट टाइप्स ऑफ इन्फेक्शंस बिकॉज दे आर इम्यूनोसप्रेसेंट उनका इम्यून सिस्टम बहुत लो होता है. So then they will die of other infections also. That is the reason.
There is another query from Shivani. She wants to ask that we take many homemade things like haldi or ginger, etc., to boost our immunity. So, is this habit good or bad?
No, it's a good habit, but you should not take anything in excess. जितने हल्दी और ये तो चलो हमारे डाइट में रूटीन में ही हम लेते हैं अदरक हल्दी सब, बट जिन लोगों ने एक्सेसिव काढ़ा पिया इस टाइम में कोविड-19 में, दे also suffered from these diseases. तो कुछ चीजें जैसे बहुत आपने मैग्नीशियम खाया, जिंक खाया, है ना, और आपने विटामिन बी टैबलेट्स लिए, सो किसी भी चीज का एक्सेस बॉडी के लिए ठीक नहीं है. जितना हम खाने में अदरक डालते हैं, चाय में पी लेते हैं, वो उसके नुकसान नहीं होता. जैसे ट्रेस मेटल होते हैं, ट्रेस एलिमेंट्स हैं, वो ट्रेस में ही चाहिए होते हैं बॉडी को. और जब आप इतना हाइपर एक्टिव होके उसको बहुत एक्सेस में लेना शुरू करते हो, then that causes toxicity. So all these taking haldi and जितना हल्दी हम लेते हैं बहुत सारा एक्सक्रीट भी हो जाता है. Not all the turmeric gets absorbed, you know, ज़्यादा आपकी एक्सक्रीट ही हो जाती है. So anything you take should be taken in moderation so that it does not become toxic.
Excuse me, Dr. Hina, मेरी थोड़ी सी क्वेरी थी मैडम. मैडम जिस तरह वी वर टॉकिंग अबाउट बी सेल एंड टी सेल, ये जो कोविड-19 है इसमें हमारा जो इम्यून सिस्टम है ये मैडम विल बी टैकल्ड बाय टी सेल और बी सेल.
बोथ. मैंने कहा भी किसी भी पैथोजन का रिस्पॉन्स है पहले, जैसे वो जो वायरस है, जैसे मैंने फिगर दिखाया था, वायरस गेट्स प्रोसेस्ड बाय द एंटीजन प्रेजेंटिंग सेल. एंटीजन प्रेजेंटिंग सेल वो एपिटोप प्रेजेंट करता है टी सेल को. टी सेल फिर साइटोकाइन प्रोड्यूस करता है टी हेल्पर सेल बी सेल्स को एंटीबॉडी बनाने के लिए. तो दोनों सेल्स हमेशा टी और बी सेल्स इसमें रोल प्ले करते हैं. अल्टीमेटली क्वेश्चन ये है कि उसमें प्रोटेक्शन हमें सेल मीडिएटेड इम्यून रिस्पांस दे रहा है या बी रिस्पांस दे रहा है. सो इसमें both of them play a significant role, but humoral immune response का उतना ज़्यादा रोल नहीं है प्रोटेक्शन में, even against viral infections. It is a cell-mediated immune response always, which is more protective. Only there are few infections where antibody plays a significant role.
Right. Second, or madam, we used to read in newspaper that being RNA virus and it's mutable. So how effective will be our vaccine which are in progress?
नहीं वो WhatsApp पे हमने भी देखा था. That is all wrong information that is given. Right. ऐसा कुछ वो नुकसान नहीं करेगा आपके जेनेटिक सिस्टम को. Immediately I am in some scientific group हमारे सर्ब के जिसमें बड़े वैक्सीन के एक्सपर्ट्स हैं. The moment it was put, so they had a big rebuttal to that कि अनपढ़ लोग हैं, कैसे RNA वायरस के अगेंस्ट डाल रहे हैं मैसेजेस. So that's all wrong, you know. They do not, जो भी वायरस जो भी वैक्सीन बना रहे हैं वो दे आर ऑल at the helm of the affairs of vaccine development, so they will not do anything which will harm the community. तो ये तो है, कुछ लोग ये व्हाट्सएप के चक्कर में बहुत ज्यादा अनड्यू एडवांटेज उठा के लोगों में हाइप क्रिएट कर देते हैं ताकि लोग घबरा जाएं. So
That's
WhatsApp,
why our principal sir was talking about.
हाँ
Thank you,
नहीं,
ma'am. Thank
तो
you. Thank
thank
you.
you so much.
Thank you, ma'am, for clearing all the queries. Now I deem it as a great honor and privilege to have been asked to propose the vote of thanks. I, on behalf of Principal Sir, college management and participants, extend my first and foremost vote of thanks to Dr. Sukhbir Kaur, who spared her time to deliver a talk on the topic Historical Perspective and Basic Principles of Immunology. I am sure that audience will have a lot to take away from the lecture. We are grateful to Dr. Ashwani Sharma, Dean, Life Sciences, for his support and guidance. My heartfelt thanks to all the faculty members of Biological Society and students. I should not forget to thank Dr. Rohit Sachdeva for his technical assistant. Thank you so much.
Thank you, Heena. Thank you so much, and thank you all for sparing so much, giving me this opportunity to present.
Thank
Share, meet you, and I would have loved to come over and then some other time
Sure.
when we are out of COVID.
Sure.
Thank you so much.
Thank you.
Thank you.